All right, so this is my final exam for microbiology. I'm just going to go over the questions that were listed here, and I did write all of my answers on a Word document, so just to make sure that I don't forget or not cover any information that I wanted covered in my answers. So if you see me reading, that's what I'm reading. Question number 1, we'll just dive right into it. What was your favorite topic covered this semester and explain why? So this semester, my favorite topic was definitely immunology and epidemiology. I enjoyed these topics because I felt they were very applicable to what we're experiencing around the world today with the global pandemic, especially with there being so much confusion and misinformation out there. This topic really helped me understand how our bodies respond to vaccines and how herd immunity can help eradicate disease. My undergraduate studies, I also did my undergraduate studies in public health, and in my last internship, I worked closely with some epidemiologists, and I just thought it was a really interesting field of study. So anyway, that's kind of why I enjoyed those topics. Question number 2 is, if you had an unknown microbe, what steps would you take to determine what type of microbe it is? Are there particular characteristics you would search for? Explain. So I have kind of a long answer here. There's a few tests and things that we can do to determine what a microbe is. And microbes can be classified by their phenotypes and genotypes. We have stainings that we could do with gram-positive, gram-negative. So I'm just going to go into that in more depth here. So in regards to microbes being classified by their phenotypes, there's different tests that we can perform in order to determine what they are. And we have biochemical tests, serological testing, and the use of the protein profile. So those biochemical tests, they are used to determine the metabolic nature of the bacterium. This helps scientists understand the metabolic pathway which the bacterium can undergo. It also has the ability to study the end products of metabolic processes. Now, serological testing is the use of antibodies to detect specific proteins or polysaccharides on the cell surface of the microbe. Specific antibodies can attach to specific cellular structures that could lead to the identification of a microbe. And then the protein profile, it basically does just that. It determines a microbe's protein profile. So it allows for quick results, and it's used a lot of times in clinical labs because patients, they need their results pretty quickly. So those kind of help determine the phenotype of microbes. Now, there's two ways that microbes can be classified based on their genotype. And one way is by detecting specific nucleotide sequences, which is done by isolating the DNA isolating the DNA from a body fluid sample. So the DNA is then denatured and combined with DNA probes, and these probes, they correspond to the gene of interest, and then they're labeled with a molecular beacon. So finally, the DNA probes will bind to that gene if it is present in the DNA sample. The only downside to this is that it can only detect one possible match at a time. So if there happens to be an organism with multiple distinguishable characteristics, then multiple probes will need to be used. So the other method used to classify microbes based on genotype is through sequencing of ribosomal RNA genes. So the RNA genes are relatively stable, allowing them to be good identifiers. So with too many mutations, a ribosome would not be able to function, and this makes sequencing them a solid way to identify the microbe. And there's also a few techniques scientists are able to perform to differentiate between two strains of viruses. So in regards to viruses, if given the task to do so, to find out what the virus is, it's good to know that viruses can differ in antimicrobial susceptibility. So that being said, taking a paper disc containing a variety of different antibiotics and placing them on bacterial agar, that's like the one thing that I still am, it's like tomato, probably not, but that's how I say it, can be an effective way to differentiate between the two strains. So After a certain period of time, clear areas will be visible around the discs of antimicrobials that inhibited or killed the strain. So this is actually what we did in our labs in regards to the E. coli bacteria when we put the antimicrobials, those discs on the on the plate. So that was a really neat lab. But another step would be to expose bacteria to bacteriophages. And bacteria can differ in their susceptibility to those bacteriophages. So the exposure of such may help in differentiating the strains. So and now scientists may also take steps to recognize subtle differences in the DNA sequence of the virus by comparing patterns of fragment sizes that are produced when the same restriction enzyme digests DNA from each isolate. So to separate the fragments, gel electrophoresis can be used as a tool. So that being said, there's also different types of staining that we can do. And that just helps us see the bacteria. So we have simple stains. So these are very basic. We have positive and negative. They're used to dye the bacteria, and the bacteria wall that has a negative charge. So the type of stain behaves differently based on that charge. We also have the Gram stain, and that's the most commonly used procedure, and it's the one we've probably talked about most in this class. So we have Gram-positive and Gram-negative bacteria, and they have a lot of differences in their cell membranes and the cell wall structure, so they can appear different colors in the stain. So Gram-negative has little to no peptidoglycan, and in Gram-negative there is a presence of an outer membrane. So Gram-negative is going to appear pink, while Gram-positive is going to appear purple. So moving on to question number 3 is, imagine you're interacting with an individual that does not think vaccines are effective. Using your knowledge of immunology, epidemiology, and microbes in general, discuss how you might converse with this individual to educate them on the virus. So I think the way I'd go about it is just talk about the facts. So just from what we've learned in this class, vaccines are the preparation of a weakened or killed pathogen and its products. However, we know now that with the COVID-19 vaccine that there's different ways that we can use the virus in order to provide immunity for the person. So like we have mRNA vaccines, which actually take the RNA and put it into the cell in order for them to make those spike proteins. So anyway, but vaccines are used to induce the artificial active immune response. So they will activate specific B and T cells to multiply, giving a person lasting protection because of the memory B cells. And as we remember from this class, those B cells, they remember each antigen that has been present in the body, and so they build up, or they attack that by producing these antibodies, which will cling on to the antigen. So the vaccines use knowledge of our immune system. So our primary immune response is typically secreting a low number of antibodies, but if exposed again, they would know which antibodies to secrete. A vaccination is the initial exposure. So it's a way to trick our immune system to trigger the primary immune response. Then when exposed again, the body can bypass the primary immune response and go straight to secreting antibodies, ultimately protecting us from the disease. In relation to herd immunity, vaccinations are going to prevent the disease from spreading within the population. Herd immunity happens when a critical portion of the population is immune to the disease, and when there are enough people immune, that infectious agent will not be able to spread because there's not enough hosts. So herd immunity protects people who cannot be vaccinated for any reason. It protects individuals who are more susceptible to the infection. So moving on to number 4, it's just a lot to talk about. Describe at least 2 positive and 2 negative relationships humans have with microbes. So I guess starting with the positive first, microbes, they help us break down cellulose in our bodies. So we're not equipped to do that on our own to digest cellulose. So for example, if I eat a nice salad or have something that contains cellulose in it, my body isn't able to essentially break that down without those microbes. So thank you, microbes, so I can have a nice salad for lunch. But that's a positive. Another one is so they can help recycle materials in an ecosystem to generate nutrients that organisms use. So without microbes, we not have access we do to oxygen, for example. And because of certain photosynthetic bacteria, the earth is provided with a lot of its oxygen that we have today. So those are some positive ones. And going to the negative, there's a lot of disease that is caused through microbes. Looking back on the world's history, some diseases that have wiped out a lot of people have been the plague, the Spanish flu. So these are some really big negatives. Microbes have killed millions of people, and through disease. On the bright side, if there's a bright side to that, we are learning more about microbes and how they interact in our bodies. So there's a lot of vaccines available out there. There's preventative care measures. Just healthcare has really come a long way since even just 100 years ago. So that's kind of a bright side to that, is that we've been able to, in some ways, stay ahead of some of these microbes that can cause us harm. On the negative side, and this is going to be my next negative, and it kind of stems from being able to eradicate and eliminate some microbes, is antibiotic resistance. And this is a negative because, as we know, those antibiotics that we take, which can help us a lot if we have an infection, those antibiotics, if we take them properly, then it can eliminate the bacteria that's causing us problems. However, we learned about mutations and how bacteria are awesome at mutating. And so with the presence of so many antibiotics in our world today, a lot of these microbes are mutating to become resistant to these antibiotics. So scientists are continually having to stay on top of this, because it can be a really big issue when these when these microbes are able to resist essentially medicine that we give ourselves to help disease and issues that we have. So that's a negative. It's kind of like a positive that's turned into a negative. But anyway, question number 5 says, many applications of microbes exist. List and describe 5 uses of microbes. So there's a lot of uses that microbes exist. So just going down the list that I have here, one is food production. So microbes play a huge role in our food production. Yeast is used to make bread, and fermentation is used to make beer. Fermentation of milk is also used to produce cheese and yogurt. We have bacteria that acts as probiotics, which benefits the good microbes found in our gut. So food production, it's a big one. I don't think I go a single day without eating some kind of milk product, so milk or cheese. So thank you, microbes. Number 2 is, I have listed that they're also used to synthesize commercial products like ethanol and biofuel, and also used as insecticides. So we can have a lot of farm growth, so a lot of produce in our grocery stores that are farmed at someone's farm, so they can essentially keep our crops healthy. Next one I have is biodegradation. I'm sorry, there are so many big words in this class. Biodegradation. So it's used to break down toxic chemicals. and some microbes can help lessen our impact on the environment. In fact, one of one of the other students in this class did a presentation about microbes eliminating plastic waste, which I think is super awesome. But so they can also be used in biotechnology. So kind of what I mentioned about farmers using GMOs to help keep their crops healthy and fight against harmful insects that can kill their plants. So I guess number 4 here, we have, yes, that's right, septic systems. So I grew up on a septic tank. We lived in more of a rural area. So the way that these work, so homes are typically, they're in rural areas, they're on these septic tanks. And it's like a small version of municipal water plants. So the home's wastewater is piped to a large tank, typically buried on the property. And this is where solid materials settle down to the bottom, and hopefully they're broken down by these anaerobic microbes. So over time, the sludgy remains need to be cleared out and often pumped out by septic workers coming to the property. But I think ours was like every 5 years or something. I don't know. It was a good amount of time. So number 5 I have listed is this one. Just I have it on here just because it was so, I don't know if shocking is the word, but kind of shocking to me to learn about initially when we were reading our book for this class. But I have fecal transplants for things like C. diff. I thought that was really kind of gross, but neat to see that this application of microbes can be used to help cure a disease. So that's just another application is fecal transplants, and specifically for C. diff is what we learned about. I don't know, I'm not familiar with any other disorders or diseases that we use that for, but just really interesting the thought process and the science behind that of introducing a community of microbes to a new community, a healthy community, and hoping that that healthy community can thrive and benefit the original community. So really interesting, kind of gross, but cool at the same time. So I just put that there because I couldn't help but think about it because it's been stuck in my brain since we read about that. So anyway, that's my final exam. I hope that I covered everything, and overall this class has been super awesome. I've learned so much, and my mind has been blown at how tiny our world can be with these microbes, and how much I didn't see before. Really, I don't see them now, but it's just really cool to know that my world is affected so much by these microbes. So anyway, thank you again so much for this class, and hope you have a great summer. Thanks.